The effect of different EDTA chelating agents on the antimicrobial activity present within a bacterial growth medium. The research question of this experiment is which EDTA chelating agent is most effective at promoting antimicrobial activity? The battle of the agents, tetrasodium EDTA versus disodium EDTA. This research is scientifically important because it illustrates the importance of utilizing the chelating agent EDTA and its variations of tetrasodium EDTA and disodium EDTA. This importance communicates through EDTA's significant purposes in current fields and industries we see today. In phytology, we see the chelating agent's use in stimulating soil phytoremediation. In medicine, EDTA provides chelation therapy to treat patients diagnosed with mercury or lead poisoning. The chelating agent is also used in the production of items such as household chemicals like synthetic detergents and industrial materials like rubber. In particular, this experimentation will be focused on tetrasodium EDTA and disodium EDTA, byproducts of EDTA synthesis that both hold antimicrobial abilities. By acknowledging these abilities, the rationale of this experiment is to test to see if tetrasodium EDTA and disodium EDTA cause different antimicrobial effects within a bacterial growth medium, and based on the rate of these effects, if data shows their presence, find out to what extent a variation of EDTA is more effective at prohibiting bacterial growth than the other, and why. The hypothesis of the experiment is that if tetrasodium EDTA gets added to a medium of nutrient agar, then the medium will have fewer colony-forming units and have a higher antimicrobial activity than that of disodium EDTA when added to a medium. This educational guess agrees on the knowledge of several needs that bacteria must obtain in order to thrive, grow, and survive, such as food, moisture, water, a high temperature, and a pH between 6.5 to 7, which is a neutral pH. Bacterial growth is often stunted by a higher concentration of sodium through the biological process of osmosis, where water is going to drive outside of the bacterium to the higher concentration of sodium so it can dilute. It is also noted that tetrasodium EDTA has more sodium cations than that of disodium EDTA, which is why the hypothesis says that tetrasodium will have a higher antimicrobial effect. The main materials of my experiment include nutrient agar, tetrasodium, and disodium EDTA, 9 petri dishes, bleach to clean up, and gloves, an apron, and goggles for protection. The main process of my procedure is that you're going to boil the agar until it's liquefied. And then once liquefied, we're going to pour it into individual bowls so we can add our tetrasodium and disodium EDTA. And then once those cool down only a little bit, we're going to pour them inside of the petri dishes so they can solidify. And then after solidify, we're going to inoculate those petri dishes with the soil bacteria. And then once inoculated, we're going to stack those petri dishes upside down and incubate them in an environment of 37 degrees, not 37 degrees, 86 Fahrenheit, and which is equivalent to 30 degrees Celsius. The physical results of my experiment is that after 10 days, disodium EDTA and tetrasodium EDTA accumulated about 2 to 3 CFUs compared to the control group, which accumulated over 250 CFUs after 3 days. And keep in mind, the control group only accumulated about 50, 20, or 30 CFUs by day 2, and the next day, over 250. Let's graph our results. The control group surpassed over 250 CFUs by day 2 to 3, with holding an average CFU growth rate of about 24.7% a day. The tetrasodium EDTA group accumulated an average of 2 CFUs by day 10. The disodium group accumulating an average of only 3 CFUs by day 10 as well. And here we can see a regular bacterial growth curve in the control group, and we can only minorly see the indentation of the tetrasodium EDTA and disodium EDTA groups. Here's a zoomed version of this. In conclusion, although tetrasodium EDTA did accumulate less CFUs than disodium EDTA, which is shown here, The data results of the experimental groups do not strongly support my hypothesis enough. This is because the TEDTA dishes only accumulated 1 CFU less than the DETA dishes. The results are not significant enough to prove that tetrasodium has a distinct antimicrobial activity that is higher than that of disodiums. So to answer our big question, which EDTA chelating agent is most effective in promoting antimicrobial activity? Both EDTA chelating agents, tetrasodium EDTA and disodium EDTA, effectively promote around the same amount of antimicrobial activity within bacterial culture mediums. So in a regular solution of nutrient agar, we're going to have water molecules and a metal ion. And once that bacteria is added to that solution, it is going to want to make a bond with those metal ions so it can carry out its biological processes and reproduce. So it's going to reproduce, replicate, double. But once those chelating agents are added to that nutrient agar, those chelating agents are then going to want to dissolve into that water. And once dissolved, it's going to want to make bonds with the same metal ions that the bacteria wants, thus prohibiting bacterial growth and prohibiting those bacteria to replicate and make colony forming units within the petri dishes. I hope you enjoyed my video. That is it. Bye.